Welcome back to biology online classes. So in the previous class I was explaining you about the meiosis, various stages, isn't it? Meiosis is also known as reductional division, and after meiosis a parent cell will get divided into how many cells? Four cells. Okay, four cells will be formed. Each will have only half of the chromosomes of the parental cell, and this is very important in case of what? Gamete formation. Okay, and in gamete formation meiosis takes place, isn't it? Why? Because the chromosome number of gamete should be only half of the original number, so that after fusion of male and female gametes the chromosome number of the parents and the offspring should be same. Understood? So for this I told how this four cells are formed, how the chromosome number are getting halved, that we are going to see. And I told you the meiosis is taking place in two different phases or events, isn't it? That is meiosis 1 and meiosis 2. Meiosis 1 takes place through different steps like prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, and in this phase only, in the first phase only, the chromosome number is getting halved in the daughter cells. Okay, after first meiotic division you can see the parent cell will get divided into two. Okay, each will have half the number of chromosomes. Understood? And in meiosis 2 what happened? The cell, these two cells, okay, formed by meiosis 1 will again get divided, so that four cells will be formed. And meiosis 2 is almost like the mitosis. So is it clear? So I told you meiosis takes place in two different steps. What are they? Meiosis 1 and meiosis 2. In meiosis 1 there are various substages like prophase 1, metaphase 1, anaphase 1, and telophase 1. And among this, the longest phase is prophase 1. And the various substages of prophase 1 is what are they? Do you remember? Leptotene, zygotene, pacotene, diplotene, and diakinesis. Isn't it? Leptotene, what is the major events? That is, the chromosomes will be visible under the microscope. Means what? The condensation of chromatin network is taking place. Okay, and that is a major event. So we have already seen that in the resting cell, or when a cell is not divided, the chromosomes you cannot see. You can see only what? Chromatin network or chromatin material. So this start its condensation. That is the major event during leptotene. We have seen that in prophase, what these are the things happens in mitosis also, isn't it? So in the initial stage of prophase 1, that is known as leptotene, you can see the condensation and compaction of chromosomes continues. Then next stage is what? Zygotene. What is the major event? Pairing of homologous chromosomes or synapses. Okay, formation of synaptonymal complex. And in this, the chromosomes that forms the synaptonymal complex is known as bivalent, isn't it? And then next comes what? Pacotene. And in pacotene, you can see the four chromatids of each bivalent chromosome will become very distinct. Okay, and you can see the sides, okay, where the crossing over takes place. Okay, you can see some sites where crossing over of non-sister chromatids of homologous chromosome. What is this non-sister chromatids? I told each chromosome will have two arms. Why? Because in S phase or synthesis phase of interphase, already DNA replication happened. But the chromosome is not split longitudinally. So each chromosome will have two complement of DNA or two copies of DNA. Clear? So like this, you can see in each chromosome there will be four arms or tetrads they'll be having, and each of this chromatids of a single chromosome is known as sister chromatids. Understood? And two sister chromatids means the if two chromosomes, homologous chromosomes, they are having sister chromatids, each one of this, and between two different chromosomes of the homologous pair, okay, they exchange the fragments. That chromatids of two different chromosome of a single pair is known as what, or homologous chromosome is known as what, non-sister chromatids. So crossing over is the major event in packaging. Then comes what, diploting. Okay, so in diploting you can see the sites where the crossover, okay, crossing over physically happens. There are some X-shaped structures that are known as what, chiasmata. Okay, and then next is what, the diakinesis. In this what you can see, terminalization of chiasmata. Okay, so these are the major events of what, prophase 1. And I told you it's a long process. And in human female, okay, the egg formation, you can see it takes lot of time. That is, by the time when a female fetus is forming in the womb of a mother, okay, from that till fertilization, if fertilization happens till that, this prophase phases means it's very long. After fertilization only the egg will get means complete, the egg will complete its prophase 1. So in means it's a very long process. Okay, so this what I told you, now you will study in the next year. So about prophase, you understood prophase 1, it's a very long phase and it contains various stages. Okay, then next is what, metaphase 1. So in metaphase of mitosis, what is the speciality, all the chromosomes, if we are having 43 chromosome, all the 43 chromosome will, sorry, 46, sorry, 46 chromosomes will get aligned in the center. Metaphase, in the meta phase means in the equatorial plane, all the chromosomes are getting what, arranged. But here the difference is, very carefully you listen, in metaphase 1 what will happen, the chromosome individually they are not making the they are not getting arranged, they are arranged in pairs. Understood? First pair, second pair, third pair, likewise 23 pairs will get arranged in a line. That is the difference between what, metaphase of mitosis and metaphase 1 of meiosis 1. Understood? The chromosomes are getting arranged in the center in both the cases, but in metaphase of mitosis, chromosomes individually they will get aligned. Understood? In the center. That is, if 46 chromosome, if 10 chromosomes are there, 10 will be getting arranged in a line, 1 2 3 4 5 6 7 8 9 10, likewise in a line. But in, if the cell with 10 chromosome, if they are undergoing meiosis, then in their metaphase 1 you can see first pair, second pair, third pair, fourth pair and fifth pair. Okay, in pairs they will get aligned. Understood? So that is the major difference. Then the microtubules from the opposite poles of the spindle, they get attached to the kinetochore of homologous chromosome. Okay, here what happened then? The spindle fibers, okay, they form and they will get attached to the kinetochore. Is what actually, centromere is the first constriction to which the chromosomes are arranged. We have studied in cell means in the cell chapter we have studied, and just above that there is a structure known as kinetochore, and its function is it gives a place for the attachment of what, chromosome means spindle fibers to the chromosomes. Okay, you can see like this. Okay, the spindle fibers are formed here, and they are getting attached to the kinetochore. Okay, then the then next phase is what, anaphase 1. During anaphase 1 the homologous chromosomes start separating. You can see here, okay, this was one pair, okay, this is one pair and this is another pair, and they get aligned in the in the form of pairs only. Okay, one pair, second pair in the meta phase. Okay, in the center. Now in the next phase, you can see at the end of the metaphase, you can see they'll get attached to the spindle fibers and they start moving to the poles. This, okay, one means from each pair, one set will move to the poles. Okay, see, you can see here, this two will move to the pole. Okay, and this two, they will move to this pole. And likewise, what happened? The chromosomes movement will take place. Okay, that is a major event in anaphase 2. Okay, splitting of the what chromosomes will not take place. Okay, in anaphase means in metaphase 1. Okay, there is no splitting of chromosomes, okay, through the centromere, but only movement of one what chromosome from each pair is happening. Okay, that is the difference. Then in telophase, okay, anaphase, then telophase, and during anaphase, where will be the chromosome? In the poles, isn't it, after completion of anaphase. And then it is telophase 1 and this opposite of prophase. What is that? The nuclear membrane, nucleolus reappears, cytokinesis will be following. Okay, and you can see a dyad of cells will form. Dyad means two cells. Okay, and the chromosomes, again they will start its decondensation. Okay, they'll form chromatin, but it won't form that extremely extended state of interphase nucleus. Okay, and the stage between these two, meiosis 1 and meiosis 2, is known as the interkinesis. Okay, interkinesis, and it is not followed by, okay, there is no DNA replication and all, as in the case of the interphase. Okay, understood? There is no it's not a long process. Okay, and here we can see a short break, okay, between meiosis 1 and meiosis 2, and then it will be followed by prophase 2. Okay, and that is also a simpler process than the prophase 1. Okay, now prophase 2, okay, or the cell is entering into meiosis 2. So in prophase 2, what all things happens? Meiosis 2 is initiated after cytokinesis. Okay, usually before the chromosomes are fully elongated. Okay, means decondensation won't be completed. Then meiosis 1 is what, it's a very simple process when compared to, sorry, meiosis 2 is a simpler process when compared to meiosis 1. Okay, and it is having more resemblance to mitosis. Which one? Meiosis 2 is more similar to what? Mitosis, not of the meiosis 1. Okay, then again in prophase, same thing, the nuclear membrane disappears, isn't it? Chromosomes again will become compact. Condensation of chromatin network continues and become more compact. Okay, then metaphase 2. So here again the chromosomes will get arranged in the equator. Okay, and here the attachment is individually, just like the mitosis 1, 2, 3, 4. Likewise, if 10 chromosomes are there, they will get aligned in what, in the center, as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Understood? So likewise they will get arranged. And then spindle fibers, okay, spindle fibers also will start attaching to the kinetochore. And then you can see in the anaphase 2, okay, the beginning of anaphase 2, you can see the chromosomes will get split through the centromere. Understood? And then they will start moving to the opposite side. Okay, anaphase 2, by the shortening of the microtubules, okay, they start moving moving to the poles. Okay, so here you see in meiosis 2, you can see the longitudinal splitting of each chromosome, but in meiosis metaphase, sorry, in the anaphase 2, 1, anaphase 1, there is no longitudinal splitting. So the chromosome number is getting halved where here. Understood? Because one cell will get only, okay, half of the chromosome and the other will get another half. And in this, once again, you can imagine mitosis takes place. Okay, so in this, what the chromosome number will be halved after the meiosis 1, and this two cells, okay, and two cells will be formed after meiosis 1, and after meiosis 2 you can see this two cells of dyad formed by the meiosis 1 will again undergo one kind of mitosis itself. Understood? And then from the number of cells will become 4. So now you clearly understood now where the chromosomes getting halved and where the cells are, means the daughter cells will be 4 in number, how it is formed. Now telophase 2, meiosis ends with telophase 2, okay, and again two groups of chromosomes, once again they will get enclosed by a nuclear membrane, then it will be followed by cytokinesis. Okay, cytokinesis will be there and it will result in the formation of 4 cells, 4 haploid cells. Okay, so this is what is the significance of meiosis. Reduction of chromosome number takes place by this, and it also helps in the increase of genetic variability. Why? Because variations are happening during crossing over, isn't it? What is crossing over? Exchange of fragments. Okay, exchange of fragments between, you can see in this, okay, diagram also. They are these chromosome. This chromosome is getting, okay, part of this, between these two there is exchange of fragments, between these two there is exchange of fragments, and you can see here they have shown this light pink one is getting a small part of the dark one, and dark one chromosome is getting a small part of light, isn't it? So like this exchange of materials takes place, so that results in the formation of variation, and variation is very very important, isn't it, in case of evolution. So this is the significance of what meiosis. So after meiosis, okay, the chromosome number of the daughter cells will be half of the parental cell, and at the same time what the chromosomes, okay, number will be halved and 4 cells will be formed. So this is the difference between mitosis and meiosis. So I hope this chapter is very very clear and you should understand it well, okay, because it's a basic thing that is there in the biology. Okay, so the diagram also you have to practice. So I will continue in the next class with another topic. Thank you very much.